Hi, folks. So, not too long ago, I covered Yelp in my startup deep dive number one. So, I wanted to go ahead and talk about startup deep dive number two. In that first one, we talked about Yelp as this like golden child, this really exceptional success story where it was a company that had a really great product, a really great approach to market, very sustainable rise, and is still a pretty significant player in its own niche. And I still use Yelp regularly. I know that they're doing fairly well as a company. It's that kind of unicorn piece that did really well over a long period of time. This story is going to be a good bit different, not only because it's a completely different industry. Today, I'm going to talk about a medical startup that is a biotech, biomedical startup, Signal Genetics. And the core focus of this story is what happens when a startup fails. More specifically, and maybe more interestingly, what happens when a publicly traded startup fails. Because this company didn't just get through the actual startup funding rounds, it went to the stock market and had a stock ticker. And that was particularly interesting. But we'll get into that. So, just to do a quick bit of background here, Signal Genetics was a startup founded in a startup accelerator connected with the University of Arkansas for Medical Sciences, that UAMS. They had an office. I don't actually know if it's still there. I think it is, called BioVentures, where the hospital and the staff and the scientists and the physicians at the hospital actually make startups based on some of the research they're doing, and then we'll partner with some other scientists and academics to go ahead and make actual startup ventures out of UAMS, which is really cool. And it helps kind of tap into the existing hospital market. It helps tap into multiple partnerships outside of that with other hospital chains as well. So when I came on as Signal Genetics, I was I forget the exact title, but I worked in the lab itself, and so saw firsthand kind of the hands-on, the medical work. And to really sort of summarize what they did, it was they would test samples of bone marrow from multiple myeloma patients that were fairly far along. So we were talking about fairly aggressive stages of a bone marrow cancer. This is a very nasty cancer, a really aggressive one, and one of the ones that has a very high mortality rate, where if you're diagnosed with multiple myeloma, there's a very low chance of survival, which is one of the really, really scary ones that it's important to understand and see if we can find better ways to tackle it. So the whole foundation of Signal Genetics was this MyPRS assay, this test that was isolating the individual multiple myeloma cells. So you would take blood samples of this bone marrow, you would filter out a specific type of cancer cells, and then we would tag them with markers could then be read in a scanner. So we'd isolate those, and then those were put into a chip that would actually be put through, and there was a proprietary system that would put any sample put through this into one of three risk groups. And they still had multiple myeloma. This wasn't a cure, but knowing what kind of risk group you were in also helped understand kind of the accuracy of your prognosis. Like, do you have as short of a time to live as you've been told? It's a very dark subject here. Um, but then also, one of the things about that is if you know you're in the third risk group, you have very little to lose by pursuing a more aggressive treatment. Whereas if you're in the first risk group, that one of the three, so one, two, and three, with three being the extreme and one being the most minor, if you're in that one group, have the highest odds of survival of one of these multiple myeloma patients. And it's one of those pieces of information that could really help get additional information about your diagnosis and then kind of help you make decisions about what treatment plans you wanted to follow through next. So it was a very small lab. There were just a handful of scientists. Right here, you've got your total employees is 25. There was administrative staff. There was the scientists in the lab. I want to say there were 10 of us at peak. There was the frontline folks bringing in the samples that would be mailed from hospitals across the United States. I think most of our subjects came from, I'm not going to share too much. I won't talk about that. But United States patients. And one of the things here is there's a really quick turnaround time. One of the challenges in general with your biomedical startup, especially one that's working with actual human samples, is they have a limited time between when they are collected to when they can actually be analyzed. So somebody had to be there live in the lab every single day, even if there was a snow day, someone had to walk. Um, we were lighter on weekends. We were able to skip over the weekends, but somebody had to be there every single morning, even if there was a power outage. Like, somebody had to be there to manage these samples, to take them in and stabilize them and make sure that they were unpacked and put in some long-term kind of storage so they weren't sitting on just the ice pack that was in the mailer for too long, so that they wouldn't be viable anymore. A couple reasons for that. Number one is the test was somewhat expensive. I mean, it was fairly expensive. And then also, to get these samples, you had to actually take a sample of bone marrow. So there was a combination of drilling into bone and then actually extracting the sample. Is a really unpleasant process. most multiple myeloma patients are fairly on in years, and so this is definitely something you didn't want to have to redo. It's one of those cases where it's like, let's do this, let's give them the best possible analysis we can within that time. So it was a very fast-paced, very I don't want to say strict or serious. It was a very fast-paced, very high-skilled lab environment that moved quickly to make sure we gave these samples the best chance of being assayed and the best chance of getting good feedback to patients as quickly as possible. Um, it's pretty extraordinary, really. And I won't talk too much about the details of the process because I'm not here to talk about that. But in terms of the business itself, so we had lab staff, we had people in an actual facility with very advanced equipment and some high advanced training to make sure to do this exact process and only this. And we had the two halves of the office where there was, again, those frontline folks that took care of the blood samples themselves and purified and filtered them out and got down to the just marked multiple myeloma cells. And then there was the higher grade analysis folks that would actually put this onto a chip and actually run the computer MyPRS analysis of it. And so you had, here are the challenges that are unique to this industry, right? So you have to have a pretty high number of staff relative to the amount of work you're actually putting in, because there are multiple very advanced steps that have to be handled by a fairly small amount of people in a small space with some expensive hardware in the machines themselves, expensive supplies in the chemicals and had to be used, expensive utilities because of all the like high-end refrigerators and such that were running 24/7. So there are a lot of things about this that have a very high startup cost. And then also you have to pay staff pretty highly because these are highly trained staff members. And then you also, it takes time to hire and train folks. Like you can't just pull someone off the street to reliably do this. Um, and so this is one of the challenges that actually makes a company like Signal Genetics not a great investment right out of the gate, because they did have the up and running lab running the sample assays. It was possible to export this and set up another lab like that, but the startup costs would have been extremely high. You would have needed a lot of investment to go ahead and get that set up. Then, of course, the staffing, training, and then any hiccups that came along the way. Like if there was a staff misfit, if somebody didn't end up accepting the role, if somebody was trained and then ended up taking another role, you really had to avoid turnover if at all possible. And because of a lot of this really worked in Arkansas, where it was based, because labor cost was a good bit lower for this level of basically everything. So the labor cost, the actual employee retention, and the utility cost to get all this up and running was significantly lower here than it would have been in, say, like New York City. And that made a big difference in terms of the profitability of the lab. And then one of the more interesting pieces here is that after the lab was set up, the staff was trained, we were running this and running the samples that we were getting, the lab was self-sufficient. If it was any other type of business other than a startup, it probably would still be here. But because startups are really built around that rapid growth, that high profitability, putting a lot of money in and then having to get it back out quickly, there was an expectation of aggressive growth that was extremely difficult to pull off. And no surprise, I talked about a lot of the challenges there, that staffing, that retention, that facility, the maintenance costs. There were enough samples and enough hospital partnerships that there was the market need and there was the product, I suppose, to support an additional lab. Like that could have gone onto another facility, but it would have been slow and it would have been very risky. And each time a lab was set up, it would be slow to set up and run, expensive, and again, a gamble. And also, while each individual lab would be self-sustaining, they would each be running at capacity, and there was no place to reduce costs. There was no place where you could aggressively increase the profit margin. There was no place to make this a more profitable business. There was very limited growth potential. And that's really where it hit a wall. And this, I mean, I was basically a lab level employee. We were fairly informed what was going on, had a lot of meetings with the executive team, but a lot of this I've had to piece together after the fact. This was years ago. This was, geez, more than 10 years ago at this point that all this happened. But yeah, so the company was founded, it got up and running, had a really successful partnership with multiple hospitals, was doing really, really well. But then the company invested heavily in an executive team whose whole role was to go and make partnerships and get more investment funding. And they were never able to get enough investment funding to continue the growth and keep the company going. And so at a point that went to a sale. Now, a lot of us weren't really privy to exactly what happened at the top or what was the break point, why it was shut down instead of the lab continuing on some level. And one of the more interesting pieces about the sale is that it was, like I mentioned, a publicly traded company. This has actually had a stock ticker, that SGNL, I believe, for Signal Genetics. And when the company was bought, so the company was sold, dissolved, one company bought the name Signal Genetics and the proprietary test, that MyPRS test. And then another company bought the stock ticker. So it was a split. It was a two-part buyout. And we were taken care of as staff members. That was really nice too. I appreciated that. The computer I'm actually recording this on actually got from the lab there as a used piece of hardware that wasn't being used, and I was able to refurbish that and polish it up. But that, yeah, it was a really interesting process, and to see it come apart like that was also really interesting. And then something that was really unfortunate and kind of really highlighted how good of a lab this actually was, how good the setup was, and how kind of precarious it was too, and some of the risks involved. The stock ticker is still going just fine. The company's still out there and trading publicly, and I have to check this to see which one. There's Miragen Therapeutics, and then actually, let me see if this is on the next page. This is an actual study that was run. I'll come back to that. There we go. So I believe Miragen Therapeutics is the one that bought the stock ticker. Yep, the NASDAQ listing. And so that SGNL is on Miragen Therapeutics. And then Viridian. Yeah, Viridian Therapeutics, I believe, is the one that bought the test, that MyPRS test. And we actually outsourced some of the staff members to go and train their staff on doing this process and running this actual test. But here was the challenge, as I talked about the importance of doing these individual steps quickly. And there's especially high degradation between when the sample is isolated, so when the bone marrow is filtered out and you have just the cells, then when they're tagged, and then from that tagging and purifying, and then getting them into the actual genetic assay where they're in that chip and they're read, there has to be a very quick turnaround time on that. Very fast. Every single delay means some of the cells die and you get less consistent results. And what the company that bought the MyPRS test is a very big company, and they had very difficult infrastructure challenges of just running these tests and then physically moving them to another facility where they sat for a little bit before they were assayed. And they never ever were able to reproduce the level of accuracy or the consistency of actually being able to analyze every single sample that Signal Genetics was. Signal Genetics was extraordinary in making sure that every sample that came in was actually analyzed. It was extremely rare for something to have to be recollected or for something not to actually get through the entire process, which is something we were very proud of. And a big part of why is that was the only test we did. We had a small facility where things had almost instant turnaround time, where once a sample was purified, it basically went straight to a chip with almost no delay. And just the bureaucracy of a large physical lab was not able to maintain that. And that really highlights one of the places where startups in the biomedical space can actually really excel, because by specializing, by having that dedicated staff in a smaller facility, they're actually able to perform things that larger facilities can't, just because of those logistical challenges of dealing with live samples. But then where the larger company was trying to do this was trying to scale it, do this at a larger volume for more hospitals and do more samples to a point where it would actually be a high growth, profitable thing for them to do. So something sustainable and a good, comfortable investment. Not being able to pull that off because of the way the facility was designed was an unforeseen complication by basically everyone. And this is old news. I don't know. They may have actually figured out how to consistently do the MyPRS test. I don't know for sure. A lot of this is speculation. But my understanding at the time was that the samples just were degrading too quickly to be run, and things just didn't necessarily work. So they did buy the rights to the test and can go ahead and continue working on it and see if they can figure it out. But it requires a very high level of precision and speed that a large scale lab can't necessarily do at the scale they were trying to do. But yeah, so there is, again, that really strong opportunity there for biomedical startups to do things that the large scale labs can't. And that's always one of the biggest advantages of startups, is being able to behave or act or perform things in a way that larger companies are just not able to do, whether it's because of how that company is built physically or organizationally, or because of just their approach to how they do things. And that's true of all startups and all large companies, where that's where your market disrupting niches are, which is also really cool to think about in hindsight. Like, a lot of this stuff I was aware of at the time, but all of it has just grown on me and become a lot more interesting over time. So yeah, it's a really interesting story. Now, I came into this saying that this is going to be the story of how a startup fails, and that more or less is true. The startup did fail. The product essentially failed. The stock ticker is not what it was. None of the staff involved originally are still involved with it, including the founders. So the company is essentially gone. And what it was doing is essentially gone as well, which is also sad. But it's one of those things where the startup model doesn't necessarily always work. Or when you are looking into a startup, you have to explore different opportunities for funding. And it's really, really, really important to examine kind of the fundamentals of what you're doing, how it's going to grow, and make sure that every single person involved in decision making fully understands what it is you have, what it is, why it works, why it's important, and how translatable it is to large scale growth, to another company. Is it reproducible? Like, what can you actually do with this thing that you have? And should you be aggressively exploring growth and aggressively expanding investment? Or should you be trying to really focus down on what your really good fundamentals are and succeed as a business on a different scale and a different way than you maybe were planning to originally? Like, this could have scaled in that same facility if that Signal Genetics lab had just moved to a larger facility and just been able to scale up what it was already doing. You would have seen a pretty significant growth in profits and ability to do larger partnerships without the loss of effectiveness or efficiency or quality of the tests. So there were ways for this to scale, but not the way that it was scaled. And it's one of those things that's a lot easier to see in hindsight, where assuming a large commercial lab wouldn't be able to replicate the results is something that we couldn't necessarily have done until we actually saw the process they wanted to follow and saw how they could go through it. So that could have been scouted out more in advance, and we could have been more aware of that and kind of avoided some of the pitfalls that we heard. But yeah, we just ran out of time, ran out of runway. Um, yeah, and I think part of that was actually the investment strategy of investing heavily in an executive team whose whole goal was to pursue that aggressive growth and extra funding. Um, and that they weren't able to get it, I think, is testament to kind of what they were trying to grow and that a lot of the fundamentals just weren't there. And this is something I think about a lot when I'm looking at other startups, kind of looking at their market approach, understanding how they're doing what they're doing. Do they have the foundation they should have? Are the people calling the shots and saying what's going on fully aware of how all of the pieces mesh together? It's very, very important that somebody at the top understands what is going on all the way down to the bottom, understand all the nuts and bolts, and understand how everything is working before making any of these major decisions. You can get lucky, and sometimes the fundamentals will translate and work to that aggressive growth policy, even if you don't fully understand it. But it's really important to fully understand it to have a very strong base fundamentals. Otherwise, it's going to be really easy to make a mistake. Maybe not the first decision you make, maybe not the second, but eventually there will be a mistake if there's any kind of lack of understanding somewhere in that process, and it exposes a crack in the whole building. So this is going to be much shorter than the last one. That's more or less it. It was a really interesting process, a really interesting story. And because this is a biomedical startup, which is well outside of the other companies I've been looking at or been as involved with, it was particularly interesting to see the unique challenges of that market in this situation. So that's it. We'll leave it there. Yeah, I'll have some links to some of these documents in this as well. And of course, I'm going to reference back to the episode 1, which is that golden child of Yelp. And then my next 2 are going to be a little bit closer to home. I'm actually going to talk about 2 current success stories and the education technology field and places that I've worked with longer and I'm more familiar with as a startup professional, as well as just as a general professional here. My time at Signal Genetics was very early on, and it was a very different time. So anyway, hope this is useful. Pardon. Let me know if you have any questions or comments about this.